All right, so welcome once again. So we'll start. Yesterday we talked. Where we stopped was B and T cell activation. Stopped at B and T cell activation. So today we'll be looking at immune responses. Moving on from to immune responses. So start with immunoglobulin structure and function. Okay. All right. So you can see that the immunoglobulin is Y-shaped. You can look at it here. See, it's Y-shaped, right? And but then it's a combination of two chains, right? A light chain and a heavy chain. So the light chain is the smaller part, that's this one, right? And then the heavy chain. So the larger chain is the heavy chain. Okay. And then so two parts is Y-shaped, two arms, one tail, right? So those are the arms and the tail, right? So that's the two parts, two arms, one tail. And then even within that structure, you can see that it has each of the heavy and light chain has two parts to it as well. Or rather, the heavy chain has two parts to it. It has the constant region, right? This is the constant region here, right? And then the it has a variable region, okay? So both the heavy and light chain have a constant and variable region, the constant and variable region. So, and this structure is very important to pay attention to because you get questions that will test all these different parts of it. Okay, so now there are also the two parts of it. So the constant region forms the FC portion, which is here. Okay, and then the light chains or the variable regions or the so you have the variable regions of both the heavy and light chain, which contribute to the antigen binding sites. And then a constant region has its own function. So the constant region functions with complement binding to white blood cells, while the variable region, which forms the antigen or antibody binding, antigen binding site, sorry, contribute to binding to the antigen. So you can see here there's an epitope. This part of it called an epitope here. So an epitope is a part of the antigen which the antibody binds to. So an epitope is is a tiny fragment or tiny part of the antigen which binds to the FAB or the antigen binding site of an antibody, right? Okay, so sorry. So looking at it now, so these different parts of it have different functions. So here you can see there's a hinge region. So some antibodies have a flexible hinge, long flexible hinge, while some have the hinge is short and rigid. And we'll see what that happens there. It affects the ability of the arms to move, the degree of flexibility of the arms to move. So there's a phenomenon or a feature of the antibodies that affects called avidity. We'll look at that in the next page, right? So some antibodies have a long hinge, some have a short rigid hinge, and that affects the ability of the arm to move, the arms of the immunoglobulin to move back and forth. And then also, if you look at this part of this FC region, you can see it's divided into two parts here, right? There's the complement binding site. So notice that the complement binding site is just at the top here, all right? And then if you look at the bottom part, that's CH3, right? So you can see there's a CH2. So up here is CH2, down here is CH3, then up here is CH1, right? So you can notice that the complement binds only to this site here, only to this side here. This is very important for you to notice here, all right? Complement binds only to that side, and then the macrophage binds to both CH2 and CH3. So the that part, the CH2 part, only complement CH2, CH3 macrophage binding. That's of the FC portion, all right, of the FC portion. So that's extremely important because you could get a question outlining the structure and they ask you which part of the immunoglobulin structure activates complement, right? And which part binds to white blood cells. So there's a difference between the parts of it that binds to complement and the part that binds to white blood cell. So that's very important. And this FC portion determines the idiotype, right? When we say idiotype, or rather, sorry, isotype. I said idiotype. Isotype. So when we say isotype, we're saying is it immunoglobulin G, A, M, E, D, any of that. So the different isotypes have different FC portion, and we'll see why this is relevant shortly. So so the immunoglobulin G has a different FC portion from immunoglobulin A, right? And the immunoglobulin A has a different FC region from immunoglobulin M, and so on and so forth. While the FAB part, right, determines the isotype, the idiotype. So when you say idiotype, that's antigen specificity. So no two antibodies have the same antigen specificity. And part of the reason is this idiot epitope what we described here, right? Part of it is the epitope that the antibody arms are binding to. And that idiotype also, the specificity of that is also located due to the hypervariable region of both the light and heavy chain arms. And then if you look at this structure as well, particularly the FAB part here, you can see there's the V here. You can see there's the VDJ portions. All right. VDJ portions. So they call this these VDJs make up the hypervariable region of both the light and heavy chain. So as we'll see, these parts are the ones that they will undergo a lot of mutations, both during the development of the lymphocyte, particularly this is antibodies, so we're talking about B cells. So both during B cell maturation as well as after it has been stimulated by an antigen. Okay. So I think those are the main points I want to mention about them there. Right. And then also notice that the antibody has three main functions, right? And these three main functions are carried out by the three different parts of the antibody structure. Right. So look at when we say neutralization. Neutralization is the antibody prevents bacteria adhering to the surface of the cell, right? So these are the cells here, and this is the bacteria. So you can see it's coated by the antibody. That's the the FAB part of the antibody, right? Preventing it from interacting with the cell so that it can go through the mucosa level, the mucosa of the body there. And so neutralization is carried out by the FAB part of the antibody, right? Then if you talk about opsonization, the second function. So opsonization is the same thing. The antibody will coat the the pathogen, right? In this case, you can see the bacteria here, and then make it palatable for the white blood cells to digest it. So the white blood cells that carry out this opsonization process of eating these foreign pathogens are the phagocytes, all right? And the phagocytes are two main white blood cells. We have the macrophage and the neutrophil. So anytime you hear of opsonization, think of, so another term for it, the opsonization is the antibodies coating the pathogen so that white blood cells can eat it. And then when the white blood cells that eat that, the process of them digesting is called phagocytosis. And the white blood cells that carry out that process are called phagocytes, right? And there are two main white blood cells, macrophages and neutrophils. So they're the ones that are able to see the pathogen caused by antibodies and swallow it up. Okay, so the next thing I'll say there is, so this is now carried out by this portion here, right? Carried out by that portion. And then finally, let's talk about complement activation. Complement activation where the the antibody will bind to the pathogen and then activate a series proteins in the bloodstream that are produced by the liver called complement proteins. All right. Just like we have a coagulation factors, you have the complement proteins in the bloodstream, and they are there to defend the body against pathogens. So the antibodies will bind to those pathogens and activate complement proteins, which can form a protein structure that will perforate the wall of the pathogen, allowing it to swell and burst. So the part of the antibody structure that activates complement is the C2, as we mentioned. Oh, let me use a lighter color. Is this part the C2? So you can see the different parts of the antibody carry out different functions. Okay, so that's that. Then looking at generation of antibody diversity. So this is antigen independent. What I mean by this is that it happens during the development of the B cell. So we said there's random combination of the VDJ, VJ that's in the light chain, or VDJ heavy chain carried out by these enzymes. Okay, RAG1, RAG2. So what are they doing? They're randomly combining the VDJ during B cell. I'll not go into too much detail since we're going moving rapidly. But this is when the B cells are in the bone marrow before they go to the lymph nodes during B cell maturation in the bone marrow. So there's some enzymes that are randomly combining. So if you remember, for those who attended my molecular biochemistry, talked about when during messenger RNA production, you have your pre-mRNA, you have the introns, exons, and then even with the exons, you have a group of proteins called a protein complex called the spliceosome that combines the exons in different numbers and that's combined different numbers of exons, and that combination will generate different outcomes. So that's what's happening here. All right. So these are the other. So that's one factor for generating antibody diversity. The second thing here is the random addition of nucleotides. So as these enzymes are cutting, the enzymes are cutting off this VDJ and joining them. You have this terminal deoxytyl nucleotide transferase. So it's an enzyme that will add sort of like glue those VDJ parts together. So how does it glue it? To add some nucleotides in those spaces that have been left by the RAG1, RAG2. Anyway, this enzyme too is very important when you come to hematology, especially with leukemias. So it's one of the enzymes that you encounter in hematology there. Then we have the random combination of heavy and light chains. Okay, so moving on, when you talk about this uh, generation of antibody dependent specificity. So now this is antibody dependent. This is when the B cell has come to the lymph node, it's in the lymphoid follicle, particularly say in the primary lymphoid follicle. It is now stimulated by an antigen. Remember we talked about B cell activation. It encounters an antigen. Initially, it's producing immunoglobulin M, all right? And now it takes an antigen, gives it to the T cell, activated CD4 T cell, via the MHC2 on its surface, binding to T cell receptor on the CD4 T cell. And then the CD4, that's the first signal. The second one is the CD40 on T cell and CD40 on the B cell. All right, let's look at that interaction here. All right. So once that interaction, both interactions happen, then the B cell is activated, and two things will happen in the B cell once it's activated. It will begin to proliferate. So that primary follicle will become a secondary follicle with germinal center, right? And then two things will happen in the B cell. Number one, the somatic hypermutation and affinity maturation. So when we say somatic hypermutation, what do you mean? We mean that the B cell will start also again begin to recombine these VDJ segments in both the heavy and the light chain. And why is it doing that? It's doing that to fine tune, to improve the specificity of the FAB region, right, for the antigen. So that's what it's doing. And that's what leads to the affinity maturation. So like they said, it's happening in the variable region. Okay, so that's what's happening there. And then you have the isotype switching. So isotype switching, like I said, what is happening in isotype switching? Initially, before the B cell was presented antigen to the T cell, it was producing just immunoglobulin M. So when you say isotype switching, it switches from immunoglobulin M to the others, G, A, E. And how does it do that? It's switching this FC portion. Remember, we said the FC part determines the the isotype of the immunoglobulin. So it will switch the FC of the IgM to the FC of the IgG or IgA. So it will switch that FC portion to switch it. So once it puts a FC portion of, say, for IgG, then to start secreting antibodies that are that are of the IgG type. Okay, so that's that. So, and both of that, mind you, are also happening in both of these processes are happening in the germinal centers of the secondary follicles, right? Okay. So now moving on, you have the immunoglobulin subtypes. So remember, we have 5 of them from the mnemonic gamed, right? Okay. So from the gamed, so we have IgG, A, M, E, and D. Okay. So you have on the naive B cell, right? Prior to activation, is expressing both M and D on their surfaces, and this act as both M and D, particularly immunoglobulin D, will act as an antigen receptor on the B cell, right? And so these cells now, like I said, will differentiate in the germinal center to undergo isotype switching, which is induced by cytokines and that CD40 ligand. So we'll come to this CD40 we're seeing it again, very important. Anything that disrupts this will lead to an immunodeficiency disorder called hyper IgM syndrome, which we'll look at shortly. So once the B cell is stimulated, it will now begin to produce other immunoglobulin of different subtypes, right? As the from IgM to IgA, G, and E, right? So, so when we talk of affinity, so when we talk of affinity so refers to the individual reaction between the antigen and antibody. Remember this is occurring in for the FAB part of the antibody, right? So that FAB part binding to the epitope of the antigen. And then when we talk of avidity, it's talking of the cumulative binding strength of an antibody. So it's talking about, so if you look at this antibody, right, just has 2 arms. So the cumulative binding strength will be of all the, if you have a multivalent, right, molecule there. So it has only 2 arms. The IgG has only 2 arms. So avidity, the cumulative binding strength will be 2. So some antibodies have more higher avidity compared to others. So if you look at this IgF, for example, you can see it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So the avidity of IgM will be greater than that of IgG because it has 10, right? While IgG has 2, which you can see here, right? So the cumulative binding strength of IgM is higher than that of IgG, even though the binding strength of IgM or IgG may not necessarily be different. They may have the same they may have the same affinity for the given part antigen, or even in some situation, IgG may have a higher affinity for that particular antigen compared to IgM, but the avidity, the available binding sites on IgM is higher than that of IgG. Okay. All right. So that talks about the cumulative strength of all the antigen antibody binding sites. Okay. So and this is for a multivalent molecule. So now looking at IgG, right? Looking at IgG. So IgG is the main one for the primary response to an antigen. Now IgM is one for I said primary, the secondary response to an antigen. IgM is more important in the say first antibody when you encounter during an immune. So this is the primary response they're talking about here, right? And then IgG is for the secondary response. The second time the B cell encounters that antigen, all right, and it's the most abundant in the serum, and it fixes complement. So remember the part of it that's the FC CH2 of the FC region of the antibody. So there are only two antibodies actually that fix complement. That's IgG and IgM, right? So fixes complement. You can also look at it as activates complement. So it carries out all the three functions that we mentioned of the antibodies: fixes complement, opsonizes bacteria, neutralizes toxins and viruses. The other Ig immunoglobulins like IgA and E, for example, don't fix complement, so they will carry out more of the other two functions. And it's the only one that crosses the placenta. So this is also important when we get to hypersensitivity reactions, where there are some disease conditions where if the IgG crosses from the mother to the fetus, can lead to problems in the fetus, right? And but the good thing about this as well is it provides passive immunity, right, to the fetus, which begins to wane after birth within like the first 6 months after birth. So associated with warm autoimmune hemolytic anemia. So this warm autoimmune hemolytic anemia is also a type of hypersensitivity reaction. We will get to that shortly, where you have the IgGs attacking the individual's own red blood cells. So you have a person, he has his IgG attacking his own red blood cells. And how do you, so that leads to extravascular hemolysis. So these red blood cells that have been tagged with the IgG will go to the spleen and be destroyed in the spleen. That's extravascular hemolysis. And this disorder has a similar presentation to hereditary spherocytosis, right? Similar disorder to hereditary spherocytosis. The only difference is that hereditary spherocytosis, Coombs test is negative there. Okay, so moving on, you have IgA. So for IgA, just think of parasites. They protects mucosal surfaces, So against bacteria, viruses, parasites. As we said earlier, doesn't fix complement, but to do the other things, neutralize pathogens and opsonize. It doesn't fix complement, as we said, and occurs in two. There's a dimer, so two IgAs joined by the J chain, and it can cross. So it's secreted in the bloodstream, crosses the epithelial surface to the epithelial layers of the mucosal membranes to get to the lumen of the GI airway or GU surfaces, right? So secreted by the secondary in the GI tract. That's this Peyer's patch. Remember, these are the secondary lymphoid organs. Protects against parasitic infestations. Uh, is the most produced, but has the least serum concentration, right? So producing all the mucosal secretions, and it is picked up. It has this fragment of a secretory component from epithelial cells. So once it's undergoing transcytosis from the blood through the epithelium and into the lumen, it picks up a secretory component that protects it from luminal proteases, right? So IgM, like I said, has a higher avidity because it has 10 binding sites. It's the one that is formed in the primary immune response. It and IgG can fix complement. Okay, can fix complement. However, it doesn't cross the placenta. Only IgG can cross the placenta. So it's an antigen receptor, like we said, IgG, IgD on the antigen receptor surface, and also secreted as a pentamer with the J chain, as you can see, just like IgA. But this one is involved in cold autoimmune hemolytic anemia. So similar to IgG. And then moving on, IgG now, it's not secreted like the others because it's present on the mature naive B cell. So it acts as an antigen receptor on the B cell, just like IgM. Then anytime you hear IgE, right? IgE, think of type 1 hypersensitivity, allergies, allergic reactions. It's also involved in some parasites as well, defending parasites and by activating eosinophils. So IgE, eosinophils, allergy, right? Type 1 hypersensitivity sensitivity reaction. Think of IgE. So involving also mast cells and basophils as well. All right, so moving on, coming down here to antigen type and memory. So this whole space here, this table is talking about vaccination. Okay, talking about vaccination. So there's so different type of antigens in a vaccine. There are some that are T cell. So this thymus independent is just T cell. Some antigens are T cell independent, some are T cell dependent, right? What's the difference? So T cell independent vaccines that are T cell independent contain antigens that will not be seen by the T cell. The T cell cannot recognize. Mind you, the only type of antigens that the T cell, we're talking specifically of CD4 helper T cell now. When they say T cell dependent, I'm talking of CD4 helper T cell. So the only kind of antigens that it sees are protein antigens. You will not see carbohydrates, that's CHO. It will not see lipids. Okay, so it will not see any of the antigens composed of that. So if you have a vaccine that contains any of these kind of antigens, right, there will be T cell independent. There will be T cell independent. Example of that is so antigens that lack polypeptide peptide component like lipopolysaccharide from LPS, as LPS from gram-negative bacteria. It can't be presented to T cell because T cells don't see anything other than protein antigens. And any vaccine that also contains antigens from that are not of protein, that are not of peptide nature, will not stimulate the CD4 T cells. So those ones will only stimulate B cells to produce immunoglobulins, and those ones are weakly immunogenic. So those are the type of vaccines that require boosters and it's boosters. So you give them now, you come back the next few months to give them again, right? So the best way to make those vaccines so that they can now be the you have to conjugate them to conjugate those vaccines, right? So that's where we come to the the T cell dependent one antigens. So the T cell dependent antigens, those ones contain a protein component, as you can see here, protein component. So the example, the diphtheria. So these ones would stimulate B cell, T cells, which these ones are better than the T cell independent. Why? Because they will create memory B and T cells, right? So for this vaccine that I was mentioning, the T cell independent, the best way to make boost its immunogenicity is to add adjuvants to it. So that's why you talk of there's a type of pneumococcal vaccine, right? This is a polysaccharide capsule, just the PPSV23 of the strep pneumonia vaccine, which contains only the capsular polysaccharide subunit, right? That one is weakly immunogenic. But when you have one that is conjugated, the conjugated pneumococcal vaccine, that's I think the PCV13 or so, that one is now more strongly, or it can be seen by the T cells, is better stimulant of the immune system compared to the vaccine that is just contains only the capsular subunit of strep pneumonia. Okay, so I think